All right. Now the DNTPs, okay, are called deoxynucleoside triphosphates. Deoxynucleotide triphosphates. What they are, phosphates. Okay? And I'm not going to draw it, I'm going to leave it up here. What they are is, hey, nitrogenous base, A, T, C, or G here, deoxyribose, and a phosphate. But there's three of them. So what it really is an individual nucleotide, not with one phosphate, but three. And we're going to learn, when you cleave a bunch of phosphates, that gives you energy. ATP, you guys learned about that in biology in 7th grade? ATP is adenosine triphosphate. Having multiple phosphates is a high energy bond. So this nucleotides that are going to add to the chain based upon their complementary pattern, right? Not going to be arbitrary, have to have three phosphates. And when they're cleaved, that gives them the energy, okay, to add on, okay, and make a bond with the 3 end. We're making a bond. Topoisomerase hydrolysis broke it. DNA polymerase is going to link the nucleotides by making phosphodiester bonds. Which means what? Water is released, and we're going to link the phosphate end of the 5th carbon to the 3rd. And by the way, we're going to start making the daughter strand. And we start working our way this way. And I'm drawing an arrow. DNA polymerase, okay? And by the way, they call it DNA polymerase 3. That was the 3rd one they discovered. So DNA polymerase 3, once the primer is on, can add the appropriate DNA, help guide. When I mean appropriate, remember, if this is an A, they must add a T here. Because the primer is offering up a 3' end of a sugar, DNA polymerase can build on that and understand the 3' end keeps being exposed. And so party people, this is important. We call this the leading strand. We're almost done. Because notice how we're adding. We are adding the DNA in the same direction the helicase is unzipping. So as the helicase unzips, the leading strand goes right with it because it's adding to the what? Adding to the what? What'd you guys just chant? Adding to the, say it at home, adding to the 3. Why? Because DNA polymerase 3 has a 3-dimensional structure that is required, okay, to get that 3 end. Okay, that's the leading strand. Let's go back to this strand. If you notice, we're making another copy using what? This parental strand. Now we're making a new copy. Okay, over here, let's talk about this strand. There's going to be some differences here. Okay, now here's the 5' end. Here comes primase. I'm going to erase this, so don't get mad at me. This is the 3' end, correct? If this is the 5' end of the old piece of DNA that we're duplicating, this is the new end, correct? If this is the 5', this is the 3 end. 3', right? What's exactly on this end? If this is the 3', what's on the other side?
5.
That's correct. That means that there's the 5' end. Can DNA polymerase 3 bind to the 5 end? No. What's on the 5 end? The phosphate end. It needs to bind to the sugar. So if we put a primer here, it can't build this way. The enzyme DNA polymerase works only at the 3' end. So my friends, this doesn't work this way. Let me show you how it works. I'm going to erase this primer. What has to happen is DNA polymerase, helicase has to unzip, primase has to move downstream. Makes a primer here. If this is the 5 prime, what is this right here? If this is the 5 prime, what's this? No, because if this is the prime, this has to be the 3 ending, correct? This is the 5 ending. And so which end can we build from? Always add to the what?
3.
Correct. Now by primase, adding an RNA what? Primer, we expose the 3 prime ending. DNA polymerase, I'll do DNA poly 3, that's the name of the enzyme, can now build to the 3. And look where we're going to go. We're going to go this way. And we're going to build a new strand that's complementary to the one that's here. It's not arbitrary. If this is an A, that's a T. But DNA polymerase can guide the DNTPs, which are the free forms of the nucleotides. But you notice something, we're going to keep going, then we're going to stop. How do we keep going? Well, we've got to make another primer upstream. This is the 3 prime ending. Primase makes a new primer. Then DNA polymerase 3 has a 3 prime ending to build to. Makes what? Dehydration synthesis, makes phosphodiester bonds, and builds the what? New sequence. This, my friends, is going in the opposite direction. We have to keep going upstream and back. So they call this strand here the lagging strand. It goes a little slower. It's lagging like your video because it has to build in the opposite direction. Think with me. Helicase unzips. This is working this way. So it's got to wait for this to unzip, go upstream, and go this way. Oh, go upstream, go this way. It's lagging. And we have, of course, because this is a biology, yes?
So does mutations happen mostly with the lagging strand?
No, we don't. I don't think we. No, the mutations occur or when we have mismatches anywhere.
Okay.
Okay, it's just that this wasn't a good. Now we find most of them. Okay, but this is where this could happen, as you can see.
Yeah.
Okay, but then we find them. But this is important. This is a fragment. This is a fragment. This is a continuous movement. So because this has to work upstream and back, it makes fragments. We have another name for this, sorry. And these are called Okazaki fragments. You can guess what nationality, okay, discovered this. Okay, so I think he won a Nobel Prize. I don't know where my Okazaki. Oh boy, I'm really destroying that. Where do I have that? I have it somewhere. Okazaki. Okazaki fragments. And these are those fragments that are going upstream. Okay, almost done. All right. Now, so you can see a new daughter strand that's complementary to the original strand is being made. And there is your new, as this goes forward, this keeps going until helicase unzips the entire thing, and now you can have 2 copies. However, if you notice though, all right, there's these these primers. These primers have to be removed, okay? So the primer right here can't be RNA. So what we have is we have DNA polymerase 1 removes them. I know it's getting busy up here, but DNA polymerase 1, so DNA poly 1 will remove the primer okay, we'll remove the primer, and then DNA, look, I removed this primer, right? Isn't there a 3' opening? Isn't there a 3' opening? So DNA polymerase 3 can attach to it and add a DNA, or what we call loose DNA, a DNTP, and bind it there. So DNA polymerase 1 removes that primer. DNA polymerase 3 adds them. Now, if you notice, we've got places where we're going to have little openings. We broke the chain. One last little enzyme connects any of those broken openings. And it's called, believe it or not, ligase. And ligase is a chemical that will make those phosphodiester bonds and link those openings. And that, my friends, is how one DNA molecule that represents unwinded in the S phase is duplicated. Obviously, a long, long sequence is a chromosome. All right? But who are the soldiers in the process? Who's doing the work here? The enzymes, the helicase, the topoisomerase, the DNA polymerase 3, what else we got here? DNA polymerase 1, the ligase. It's all controlled by your proteins. So what do you think happens? How do we know we get to the S cycle? We got to make sure those proteins are what? Made. Okay, so there's some kind of checkpoint that we'll talk about that allows these proteins to do their thing. All right, a lot going on here. We're not done with it, but you would have never understood what I said today if I didn't have you connect those piece of papers. The 3', the 5'. And this is not something I'm making you know because I want you to. You have to understand this process for AP Bio or biology in general. But where does it occur? S phase. So for those that are home, okay, you can chime out. You guys can take a picture of that and keep it, or you can just take this picture.
Multiple pictures.
Okay, awesome. Okay, I could post this as well. This is what I used last year. Okay, but just drawing it. Hopefully, you could see what we did here. Okay, all right. For those of you at home, you could start the homework. The homework is on 3 lectures by college professors who talk about exactly how these DNTPs, the free forms of DNA, actually link. What you're going to learn today is why it builds from the 3. Professors are going to say 5 to 3 direction it builds. It adds to the 3, adds to the 3. Okay, take your picture as your notes. I really believe in whiteboarding. If we were a full class, we would have done that. We would have ranked them, we would have measured them, had fun in any case. All right, so hope you can see we're doing micro, macro. I'm trying to keep you involved. All right, so those at home, you're done with me today. Those that are still with me, okay, we're going to open up our DNA activity and see if we can finish this up a little bit. Yeah, you too, you too. In fact, I won't be in school tomorrow. Okay, tomorrow I'll post it. You're going to work on the brine shrimp lab. Okay, you guys are going to write a background. I want it peppered with evolution, as much genetics as you can. All right, you can add to all of this. Hey, what allele does the brine shrimp have that made him more successful? Okay, in salt water. There must have been some mutation on some chromosome that codes for some new protein that catalyzes a set of reactions that makes it somehow. So in my backgrounds, I'd be going after exactly how do brine shrimp survive in saltwater conditions. And why was that an evolutionary type of adaptation? It gave them what? Reproductive fitness. Survive better. Why did they survive better in salt water? Okay? All right.